Hello and welcome to this lesson for Advanced Higher Biology. We are still on Unit 1, Cells and Proteins, and we're working our way through Key Area 2, the Proteins section. And this is the second part of Part C, which is going to be looking at ligand binding. So up until now we have looked at protein structure, how amino acids and R groups can influence the structure of the protein, and we've looked at primary, secondary, tertiary and quaternary structures of protein. What we're going to be looking at today is something called ligand binding before we move on to look at phosphate binding. So for today we need you to learn how additional molecules can bind to and affect the protein. You need to be able to state what is meant by a ligand and explain how one might bind to a protein. You need to describe how the ligand binding might affect the protein shape, and you need to define what we mean by an allosteric enzyme and the impact of different interactions between sites on a protein. So to start off, we need to consider what a ligand actually is. So it's any substance which can bind to a protein. It's usually specific to that protein, and it has a shape which is specific to the binding site of that protein. So for example, if our protein was an enzyme, its substrate would be considered the ligand. So it's important to realise there's a slight difference between when we talk about ligands and when we talk about a prosthetic group, which we mentioned in the last section. A prosthetic group tends to be a permanent binding to the protein, whereas when we bind a ligand, that's often a temporary binding for that ligand to affect the protein in some way. Obviously, that's not always a hard and fast rule, but it's usually a good way of thinking about it. The other main difference is that a prosthetic group is often needed for the protein to be able to function, whereas with a ligand, that's not usually needed for the protein. It may influence the protein and the protein may affect it, but it's not usually needed for the protein to actually carry out its function. Now, the way a protein will actually bind to a ligand really relates to the R groups. So it's the R groups that are present on the protein chain determine which specific ligands it can bind, and that usually means using either shape or charge. So as you can see here, we've got our unfolded protein, which is then folded to form a specific binding site, and those little coloured dots that are sticking out are intended to indicate the different sorts of R groups that might be present. So the binding sites on these proteins normally have a complementary shape and chemistry to the ligand that they bind to, and it's usually only the R groups that are not already involved in the folding of the protein that are free to bind to the ligands. So obviously there are other R groups present on that protein chain, and they've been involved in folding the protein into a specific shape, leaving those R groups free in order to bind the protein. Now, since the protein folding is dependent on the interaction of all of the R groups and prosthetic groups, for that matter, as a whole, anything that impacts on those interactions will impact on the folding, which will in turn impact on the function of the protein. So what that really means is, if you consider the induced fit model of an enzyme, they undergo something called a conformational change. So they have a change in their conformation or their shape as they bind their substrate. So we know about the induced fit model, and we know that when a substrate comes along and binds onto the active site, the enzyme changes shape ever so slightly in order to fit around its substrate a little bit better. The reason for that is that ligand, that substrate that is binding, it is affecting the R groups that it binds to. So as it affects those R groups, maybe changes their charge ever so slightly, that has an interaction with the rest of the R groups of that protein and consequently affects its shape. So the ligand binding into that binding site influences the R groups of the binding site, which influences the R groups of the rest of the protein, which will affect the way the protein is folded and overall affect its shape. Now we can see this really clearly when we look at something referred to as allosteric interactions. Essentially what we're looking at is how there are interactions that can occur between sites that are actually quite far apart. For example, if we look at this particular enzyme, it's referred to as an allosteric enzyme. It's got an inactive form and it's got an active form. When the substrate binds to one active site, that increases the affinity of the other active sites for more substrates. Essentially, what it's done is that substrate has bound into one of these sites. It has affected the R groups around here. That has consequently affected the interactions across all of the entire protein and the way it's folded, and has affected the shape of the other subsites here and here. So it makes it more available. It's changed their shape so that they're more available to bind more substrates. And that's quite important because obviously that gives us the idea that those enzymes are going to get more active if we have small changes in substrate concentration. So the more substrate that is available, the more likely it is we can convert our enzyme into its active form rather than its inactive form. And that's referred to as being an allosteric interaction, where the binding of something on one area of the protein affects a second area which is quite spatially distinct from it, it's quite far away. Now this can also happen in allosteric enzymes, and that's one which will change conformation after the binding of a modulator. And these modulators will bind at the secondary site, which is the allosteric site. So that's separate and distinct from the active site of the enzyme. When we bind the modulator, the conformation of the active site will change, and that will alter the affinity of the active site for the substrate. Now, the effect of this binding can be either negative or a positive. When we have a negative modulator, when it binds onto the allosteric site, that's going to alter the shape of it, but generally distort it or change it in a way such as that the substrate is unable to bind to the active site. And that can be quite important if, for example, the allosteric inhibitor or the modulator that is binding onto the allosteric site, this might be one of the products from this reaction. So it may be able to create some kind of inhibition so that the enzyme stops being active and stops producing more substrate. As obviously the cell runs out of product, that would be released from the active site, from the allosteric site, and allow the active site to return to its original shape and continue to bind the substrate for the reaction to occur. So we also have the converse, which is positive allosteric binding. So that's where you've got an allosteric activator. Essentially, the modulator is acting in a positive fashion. It's converting a distorted active site into the correct shape in order for it to bind its substrate. So binding onto here is affecting the R groups, which are affecting the folding of the protein, which is affecting the overall shape, and then giving us the opportunity for that enzyme to bind its active site. Now, as a result of this, we can see something called cooperativity in some proteins. And one of the best examples of this is the binding and release of oxygen by hemoglobin. So as you can see, if we have our hemoglobin molecule here and it's ready to bind oxygen, the more oxygen molecules that are bound by that hemoglobin molecule, the greater the space for further oxygen molecules to be bound becomes. So we have an interaction from the binding of oxygen to this group here is going to influence the shape of these groups, making them more open to binding more oxygen and so on. So essentially changes to one subunit after binding oxygen starts affecting the other subunits and allows them to bind oxygen more easily. Now, this also has a converse effect. We have something called the oxygen dissociation curve. And essentially what we're looking at is the fact that a decrease in pH or an increase in temperature lowers the affinity of hemoglobin for oxygen. So if we look at this graph, what we're looking at here is the partial pressure of oxygen in the blood versus the oxyhemoglobin saturation. So essentially oxyhemoglobin saturation is just how many oxygen molecules are being bound to a hemoglobin molecule. So if we're at 100%, then all of our hemoglobin has bound all of its potential oxygen molecules. And if we're at 50%, then we might maybe have our hemoglobin has only bound 50% of the possible oxygen molecules that it could. So essentially this blue line here is representing what we would consider normal physiological conditions. So as we have a higher pressure of oxygen, we're going to increase the amount of oxyhemoglobin saturation. However, this red line here gives an idea of, it really represents the effect of decreasing pH or increasing temperature, whereas the green line is the opposite, what would happen if we increased pH and decreased temperature. So if we consider, let's consider 50 down here, we've got normal physiological conditions. If this was our normal partial oxygen pressure here, we've got approximately, under normal conditions, we have approximately 80% of hemoglobin is saturated. If we decrease our pH or increase our temperature, we're going to drop that down to nearer 75%, 70%-ish. So the change in this saturation is because hemoglobin has had a decrease in its affinity for oxygen, so it's less likely to bind oxygen when we have a decreased pH or an increased temperature. So that's particularly useful because if we have tissue which is actively respiring and using up oxygen, it will essentially, it will have a higher temperature, generally speaking, and will also be producing more carbon dioxide, which dissolves into carbonic acid and therefore reduces the pH of that tissue. So as the more the tissue respires, the greater the increase in temperature and the decrease in pH. That reduces the affinity of that hemoglobin to bind oxygen and releases more oxygen out to the tissue. So this is referred to as the oxygen dissociation curve, essentially the fact that oxygen starts to be released from hemoglobin when we have a decreased pH or an increased temperature. And essentially that's most likely to provide extra oxygen for respiring tissues. So from that you should know about how additional molecules can bind to and affect our proteins. That a ligand is a molecule which can bind to protein and tends to bind to a specific site, the shape of which is created by R groups. And that when that ligand binds, because of the interactions between the R groups, it may have a significant effect on the protein shape. And an allosteric enzyme is one which is a secondary site, and that will be regulated by positive or negative modulators. And we have different interactions between different sites, specifically cooperativity, and that can be seen in oxygen and hemoglobin. Thanks very much for paying attention so far, and let me know if you have any questions.